Okay, well, it's 3 past, so we might make a start, and I imagine, considering the early hour, a few people will trickle in as we get going. But slow roll.
They're probably not through with brekkie yet, right?
Well, so you've got to have a good cup of tea here before you sit down to a session like this at this time of day. So, anyway, for everyone that's joined us so far, welcome to this webinar with Dr. David Johnson. It's a pleasure and a privilege to have you with us, and we're really excited.
Don't forget
and Hui-Chun there. Yeah, so I'm just going to just do a couple of formalities and then pass over to David. My name's Sam Lang. I'm the manager of Quorum Sense at the moment, so I've been organising these webinars on behalf of the group, and we've run probably 7 or 8 in the last couple of months on a range of topics, mostly talking with arable farmers on their experiences from the season and what they've been learning and what they're thinking about going forward, and which has been really cool. And you can find that kind of content on our YouTube channel, which is Quorum Sense NZ on YouTube. There's a couple of grazing discussions on there as well, and a whole lot of videos from previous events that we've run. So if you're new to this and you like what you're hearing today, feel free to dive into that content in the future. And we are making, we will make this, I am recording this webinar, and we'll make it public on that YouTube channel as well a few days later. So if anyone's uncomfortable having their name mentioned or anything like that, it hasn't come up before, but feel free to just email me afterwards, and we can edit that out. It's contact@quorumsense.org.nz. And otherwise, yeah, so just a quick background. Simon Osborne, whose house I'm at this morning, and I, an arable farmer here in Leeston, we built 6 of the Johnson-Su compost reactors that we'll hear about today just over a year ago. And we had a whole lot of sort of waste material, and I chipped up a whole lot of fresh willow and that kind of stuff, and we had a nice big machine that we managed to pull out of the shed to do the job, which made it quite fun. Yeah, so they're just getting on a year old, and theoretically getting to the point where they're starting to mature, and David will be telling us a bit more about that. But we did that because we're fascinated to explore this, how we can continue to extend, what this, I'm going to show my lack of knowledge here soon, but what role this kind of really high diversity compost can play, and really curious to see how that compares to a vermicast product, for example, or the kind of biostimulants and bioinoculants that other farmers are putting in that are already commercially available. And from what we've been able to discern, the Johnson-Su composting process possibly produces the highest quality inoculant that certainly that I've come across, and so just really fascinated to learn about I suppose your story, David, and how you got to where you are, and how the Johnson-Su came to be, and what the understanding behind it all works. And I know you're going to be talking to us a bit about that. And yeah, we're hoping that we'll have a good amount of time towards the end for some Q&A. So if you haven't used a webinar session before, there's a Q&A box down at the bottom of your screen, and if you type your questions in there, once David's finished his initial presentation, I will be looking through those questions, and I'll ask them on your behalf, and hopefully we get through them all. And so with that in mind, I should also mention we've got Dr. Gwen Grellett from Manaki Whenua, who's a great friend of mine and has been really, really active with Quorum Sense over the last couple of years, and increasingly involved, and I work with her a lot. So she's going to be co-hosting with us this morning. So thanks, Gwen. But I think for now we'll pass over to you, David, and thanks very much for being here.
Well, thank you for inviting us. Much appreciated. Well, as far as how this started, it was all about dirty laundry. My wife, I had received a project at the university to do something good with dairy manure. And I was in the old methodology, turning windrows. And my wife got tired of me coming in with dirty clothes, and it was her suggestions and intuition that put us onto this style of, static compost bioreactor. And it is all because she didn't like to do the dirty clothes. So I will switch into the presentation mode here. So I guess first what we need to understand that microbes are the backbone of every organism and ecosystem on this planet. They're critical for every life form on this planet to function and survive. Us, we're sitting here with about 10 trillion human cells. We have about 90 trillion microbial cells. So we're outnumbered almost 10 to 1 on cell count. You also have about a one quadrillion resident viruses. So you are not alone. You have about 20,000 human genes, but you have 2 to 20 million microbial genes. So you are less than 0.1% human, and the rest of you is mostly microbial. What do they do for you? Just about everything. They digest your food, they do nutrient generation, vitamin synthesis, cell renewal, they control your appetites and cravings. We used to think they were about 60% of our immune system. We're now seeing more like 80%. They detoxify carcinogens, prevent allergies, and they also turn on and off genes that regulate brain development, anxiety, depression, even autism and arthritis and emotional behavior. So what happens when these microbial communities are disrupted through either administration of antibiotics or diet-related issues? Just about every autoimmune disease that we have is related directly to your microbiome, your stomach microbiome. You can see inflammatory bowel disease, IBS, cardiovascular disease, diabetes. Several of these are directly related to your microbiome. We're seeing since 1950 a dramatic increase in the incidence of these immune disorders, from type 1 diabetes to asthma to multiple sclerosis, even Crohn's disease. Crohn's disease, they've seen in people that have suffered decades. And some people can die from this. They'll have surgery to remove parts of their intestine. They'll take more antibiotic regimes. But they found that if you come in with a healthy donor with a fecal matter transplant, that these people that have suffered for decades, in some cases, can be cured within 24 hours. It's a dramatic turnaround once you get the right biology going. This is kind of a look of the diversity of our stomach microbiome. You can see here the USA is quite a bit behind people from Malawi and Tanzania as far as diversity. You also can see the influence of diet. What they did on this experiment with mice was they removed fiber from the diet of the mice. And you can see there's a decrease, generation after generation after generation, losing that diversity. Well, you say just bring it back. Once you bring the fiber back, they did not see a return of the diversity in that community until they came in with fecal matter transplants, bringing the biology back into that system to regenerate it or repair it. So if this works in the human microbiome, can it work in the soils of agriculture? We're seeing that potentially it can, but first we need to understand a little bit about the succession ladder and how nature has populated this planet. Going from a bacterial dominant soil up to a fungal dominant soil, we see a change in plants from true bacteria and protozoa, weeds, grasses, vegetables, row crops, shrubs, vines, deciduous trees, and conifer and old growth forest. We also see along this change from bacterial dominant to fungal dominant an increase in the productivity of these systems from about 90 grams of dry biomass per square meter up to 2,200 grams of dry biomass per square meter. This is an image of fungi. There's a film of water on the fungi, and these are bacteria that are crawling along that film of water. This is real time. So this is Pseudomonas. So they are very important in the soils, but even more important are the fungi. The fungi are the logistics and communication of this system. But that's the first thing that we wipe out with the management that we have in agriculture. Well, nature used cattle and bison to form the deep soil carbon profiles in the Great Plains in the US. The bison were kept in tight herds by the predators that moved them along regularly. They only grazed about 40% of the forage that was available. They would deposit their dung, their urine, their saliva, and their dung would be rolled up into small balls by dung beetles and placed in the ground to basically compost in place. It was a whole system that developed, and it led to the buildup of carbon, very rich carbon soils that, well, we've been mining for several years. So what we are saying, can we mimic this system in agricultural lands? Can we duplicate what's going on? Because this was like an inoculation event that went across the prairie. Every time the bison would go across, they would inoculate an area, and that would regenerate that system. My wife and I developed a composter. You can see here, it's very simple. It's a static composting process. You can see there's tubes in the center of the pile. Those tubes are only in there for about 24 hours after you build the pile. They offer air columns that allow air to flow up through the centers so this stays aerobic. So what you need to maintain in this composting process is about 70% moisture content on the material. There's no turning involved, no disturbance. Hence, the fungal community can proceed and go forward, where in windrow composting, it's disturbed every time they turn it. It's like they have to rebuild their whole system every time you turn it. It's aerobic with air columns through the center of the pile. You add worms after the temperature goes down to about 80 degrees. I think that's about 26, 27 degrees C. And it takes about 1 year to reach maturity. What we see in this is it produces a fungal dominant and biologically diverse compost. This is what the texture of the compost looks like when you're done. It's like a clay. You can squeeze it between your fingers in a ribbon, and it's the whole thing when it's done looks like a dense, dark, rich chocolate cake. We looked at some of the bacterial community at the phyla level as you go through the composting process, from 159 degrees here down to about 60 degrees. You notice an ebb and flow of the different phyla in this system. Some of them start very strong at the higher temperatures, but they're practically non-existent in the end. Others, like the proteobacteria, relatively strong. They fade away, but then they come back in the system to be a dominant community member. This is an example of 2 compost samples made 10 years apart with totally different starting materials. The top one is a dairy waste along with wood chips, 3/8 and yard trash. And the bottom one here is just all leaf compost. But you can see that there's a striking similarity in the structure of that community. We think it's more the process than it is what materials that go into the system. And you can see at 4 weeks, there's about 23 species in the top 80%. At 22 weeks, when many composters say that their compost is done, there's about 57 species. At 60 weeks, after that 1 year mark, we see 99 species. So you have a 4 times increase in the biodiversity of the system as you allow it to mature. And remember, diversity is the currency of survival on this planet. So we think that's part of the reason this works so well. When we started looking at the compost, we compared it to 8 other composts in our area. These are our 2 here on the left, looking at the growth compared to the nitrogen content of these composts. You can see our compost pretty much had very little to no mobile nitrogen. And yet the plant growth in ours was over double any of the other compost available here. Phosphorus as well. You can see we did not have that much phosphorus, but we had excellent growth. And potassium, again, some of the lowest numbers, and yet none of the nitrogen, phosphorus, and potassium correlated to plant growth. We were thinking, okay, organic matter. But again, there was a very poor correlation to organic matter. Now, organic matter is important, but it's not the greatest correlation to what you see as far as plant growth. What did come up was fungal to bacterial ratio. Very good correlation, as you can see, relative to the plant growth that we observed. That started another experiment in the greenhouse, growing chili. And as you can see, the chili leaves on some of these treatments were pretty awesome. That's not a normal size for a chili leaf. It would be about a quarter of that in the field. But looking at, we wanted to see where the carbon and where the nitrogen in these systems was partitioned. So we mixed up a ratio between the soil and the compost to get a fungal to bacterial ratio, starting fungal to bacterial ratio of 0.04. And we ended up at a 3.68 fungal to bacterial ratio on the top end. Now keep in mind, this is the same biology all the way through. It's just in different ratios of from a sand mixture and a compost mixture. We wanted to see, okay, where does the carbon go? How is it partitioned in this system? We saw in a bacterial dominant soil, low carbon soil, there was only 3% of the carbon in this system actually devoted to plant growth. The rest of it went into new soil carbon formation, respiration carbon, or total new carbon, or total nitrogen assimilation, and the carbon used to do that. The carbon, it's very expensive carbon-wise to fix nitrogen. It takes about 12 units of carbon for every unit of nitrogen fixed. So we looked at that as we moved up through the fungal bacterial ratio. So first of all, let me tell you, the red is the amount of carbon that went into the fruit. The green is the amount of carbon that went into the shoot. And the brown is the amount of carbon that went into the root. Gray is new soil carbon. Blue is respiration carbon. And again, the orange is that total new nitrogen carbon related to that. As we moved up from fungal to bacterial ratio, we'd see increases in the partitioning of that carbon into the plant structure from 17% to 21%, 30%, 37%. And finally, in our last treatment, we saw 56% of the carbon that was fixed by that plant go into the plant. So where we are in agriculture right now is right here. We're bacterial dominant, we're low carbon. We're sitting at about 11% efficiency. And we see that the potential, according to this, should be a 5 times increase in what we're seeing in our system. So does this work in the real world? What I'm talking about is a biologically enhanced agricultural management, or BEAM process, where we're focusing on the development of the microbiology in the soil, but not only that, but the system in general. That's what evolved on the Great Plains. It was a system. It involved the predators, it involved the grazers, it involved the insects, the birds, the grasses. All of these work together, and that's what we're trying to emulate in our agricultural system so that we can start to increase our productivity without all the damage that we're doing to our environment. First of all, you need to see that agriculture is not very efficient. Our cultivated land only does about 600 grams of dry biomass per square meter per year. Nature, without fertilizers, without watering it, without weeding it, you see about 2,200 grams. So we're very inefficient right now. We're seeing with a BEAM transitional that we can match that 2,200 grams. We're seeing that as we move into a more advanced BEAM soil, we're seeing up to 3,200 grams. So 5 times increase from what the current productivity is. We started out spreading this compost at about 400 pounds per acre, or 450 kilograms per hectare. This is what it would look like on the soil. It's basically just a dusting. It's not a nutrient amendment. It is a microbiological treatment that you're doing to these soils. This is what it looked like after 1 year of BEAM. On the right-hand side, we'd had a winter cover crop, a summer cover crop, and this was our next winter cover crop. The control, as you can see, only produced about 50 grams per square meter. And the BEAM was already 250 grams per square meter after the first year. So that's a 5 times increase. 7 years later, after we continue to grow cover crops and put them back into the soil, we see 1,141 grams of dry biomass per square meter. We saw improvement year after year after year. This was, as I say, year 7. That's about 5 tons per acre, or 387 kilograms of nitrogen per hectare on this system. But we also saw a 5 times increase in the biomass from that after that first year. So there's a lot of potential here to improve the productivity of these soils with the biology. This is a summer crop, cover crop after that. It has about 5 species in it. It has black oilseed sunflower, it has sesbania, it has soybean and millet. And you can see June 23rd, it was about 4 to 6 inches tall. A month later, it was about 5 to 6 feet tall. This was 17 days later, it was over 7 feet tall. And normally, black oilseed sunflower do not grow this tall or have this big a head. This picture on the right is sesbania. It's a crop that's native to southeastern California and southwestern Arizona. It's a legume, and it also associates with mycorrhizal fungi. The first season we grew this, it was about 4 feet tall. The second time we grew it, it grew 6 feet tall. And this is the third time that we grew it, and it was 12 feet tall. So each successive year, we would see improvement in these soils and their ability to grow forage. We also looked at what the biology can do in a non-legacy soil, a soil that had never experienced agriculture. As you can see behind me here this is our environment. We're in a desert area. It used to be grasslands, but improper grazing left us with this. It's very dry, very barren, sandy soil. What we wanted to see is just what the biology could do as far as productivity. On this one, on the cover crops that we grew, we would cut them and haul them off and then plant again, taxing the system as much as possible just to see what the biology could do. Our first season on this winter cover, we produced about 800 grams of dry biomass per square meter. This is the second season. We produced about 1,608 grams of dry biomass per square meter. This is the third season, 2,190 grams of dry biomass per square meter. If you remember, the most productive ecosystem on the planet was about 2,200 grams. We were very close to that. And realizing that this crop is planted October or November, it only gets a couple inches high during the winter. It wakes up mid-February, and we harvest it at the end of April. So there's about 10 weeks of growth in a time of the year that there's no other crops growing here, that you produce as much biomass in 10 weeks as the most productive ecosystems on the planet do in a year. So there's a lot of potential here for improving agriculture and improving productivity and profitability. This is what it looked like on the winter covers, season by season. Without the inoculation, we're doing 492 grams at the beginning compared to 820. You can see as you go through, you keep getting these increases. That data by years is here, looking at, the cover crops are essential. But bringing the biology into the system kickstarts it, and you get half again the productivity over just the cover crops. You're seeing at the final year 3, we're seeing 3,186 grams of dry biomass per square meter. And that's 31 tons of dry biomass per hectare. We were seeing over 872 kilograms of nitrogen per hectare per year fixed. So we also see from 492 to 3,186, we're seeing a 5 times increase in net primary productivity as we bring this biology back into the system. This is what the soil looked like when we were starting. No A horizon, just sandy soil. This is the control. We saw a 3-inch A horizon develop. And this is the compost treated. We saw 7 inches of that A horizon develop in that 3.5-year experiment. Of course, I'm a molecular biologist. I like to look at the biology on these systems, and we could see, comparing the desert soil that we started with to our control, to our compost treated, over 23 different phyla had more population in the compost treated than in the control or the desert. So we do see that we are restoring diversity in this system as we inoculate with this microbial amendment. Again, diversity is the currency of survival on this planet, and we see that as the key for what you need to be doing in your soils. We saw in a metagenome analysis, looking at all the species, we found about 2,750 species of bacteria in this system. There was about 193 that came to the top and correlated with the plant growth we saw, or either that or had populations from 10 to 100 times what the norm was. Now, these organisms do all kinds of different things. We saw the nitrogen fixation, both free-living and symbiotic nitrogen fixers. We saw nitrogen and carbon cycling microbes, which are critical to this system. It's that flow of energy in that system, and it's the nitrogen cycling and the carbon cycling that make this possible. It's like an economy. If you don't have a certain velocity of money in a system, it will collapse. Well, it's the same thing with carbon and nitrogen in the soil, and that's their money that they deal with. And it's that carbon and nitrogen. We saw organisms that would do metal oxidation. We saw ones that would do phosphorus solubilization. And for the most part, those soils in agriculture have 40 to 50 years of phosphorus in the soil. It's just not available to the plant because of the soil physical chemistry. Well, microbes change that. We saw also antibiotic producing microbes that control the community structure. We saw biofilm and quorum sensing microbes. We saw microbes that could degrade pesticides, and we also saw phytohormone producing microbes. We saw microbes that actually produce cellulose in the soil, helping to build soil carbon up. They were a dominant member. We saw this particular Frankia species does 15% of the world's biologically fixed nitrogen. This next one here was radiation resistant. It can be bombarded by gamma rays. It can have its DNA broken up. It can reassemble itself and go on. So these microbes have some amazing talents. One of these is quorum sensing and biofilm formation. And it's interesting. This is a plug for the organization. But it's very critical. And quorum sensing is when you have a community of microbes that start to work together. That can express genes as a community that they can't as a single individual. Quorum sensing is involved in symbiosis as well as virulence. That's how we get sick. A certain number of microbes populate us, and they work together as a community, and they start secreting toxins. Well, it can work for the good as well in symbiosis. And what we noticed in this system, that these down here are just regular genes that are translated for what we call housekeeping functions, which are critical. It's the basic function of a cell, like ribosome formation here. Ribosome makes all the proteins. And this baseline level, what we saw were only two proteins that were expressed at from 6 to 10 times the rate that the housekeeping genes were. So it was interesting to see that both of them were involved with quorum sensing. So I think that's what we're trying to do to ourselves, is bring back that population of microbes and allow this process, the quorum sensing process, to start to develop. And that's why I think we're seeing all the benefits to this system. So what are the benefits of regenerative ag? We're seeing the ability to increase carbon at a much higher rate than most other researchers are seeing. In West, Nigley, and Mall, they're seeing only about from 0.5 to 0.7 tons carbon per hectare per year increase. We were seeing in our soils with the beam approach, 10.7 tons per year of carbon increase. That's about 0.25% soil carbon increase per year. And we see the potential to do 0.5%. And what was nice to see is it was a pretty good chart that we observed at that 10.7 metric tons at 0.25% that we saw the increase. We'd see what Gabe Brown had tried. And he had tried for 13 years with no-till, crop diversity, cover crops. His carbon accrual rate was only about 0.1%. Not until he reached about 3% soil organic matter. he stopped using nitrogen fertilizers, and he started implementing multi-species covers. And finally, you can see after livestock integration, this thing just went crazy. But he was averaging about a little over 0.5% carbon increase per year. And that carbon is essential. That's an energy resource for this microbial communities. And it's how they function, like energy is for us. This is his pastures. We visited him this last July. He had gotten about 5 inches of rain for the last 3 years in a row on this grasslands. Now, normally 5 inches wouldn't grow much, but this grass was over the top of the hood of his truck, and he has a 4x, so it was about shoulder high out in these fields. And this is what his soil looks like. This is the aggregation that he's built up carbon, significant carbon to depth, and he has a system that's very resilient and that can produce on very little rain. What we also noticed in these soils, as you went, this is back to the greenhouse trial. As you went from that bacterial dominant to fungal dominant, you see the respiration in this system. This is all related to carbon use efficiency in the soil. But, excuse me, we saw an 18 times increase in beginning soil carbon mass as we moved up this ladder. We also see a 5 times increase in microbial biomass. Now, most of the researchers are going to say if you see these type of increases, the carbon respiration should be exponential, but you see it level off. As the system gets more efficient in the usage of carbon, we only saw a 4 times increase in soil carbon respiration mass in this system. That's another reason that you can start to build up carbon faster in these soils, is that the microbes are using the carbon more efficiently, making more microbes for the most part. This is Will Harris's in Georgia. This is his 5 different plots he has at different years in the regenerative grazing, looking at the respiration in this system as he moved up from 0.25% carbon to 7.9% carbon. You can see again, he has the same exact curve that we do. As he built the carbon up in the soils, his respiration did not increase linearly or exponentially, but it plateaued. He had a 10 times increase in beginning soil carbon mass, but only again a 4 times increase in carbon respiration mass. Looking at this just a little different, in that bacterial dominant soil, 55% of the respirable carbon in that soil was respired. And yet, when you move over here to a fungal dominant system, we see a 10 times increase in beginning soil carbon mass, but only there's a 4 times decrease in the relative soil carbon respiration. So we're improving carbon uses of efficiency in these systems as we bring the biology back. What does this do for water? That first 1% increase in soil carbon is going to give you a 5 times increase in water storage. And this is critical that you're able to store the water and use it more efficiently in these systems, especially in dryland farming. We also see in our food supplies that we are losing the essential minerals that we need for our health. This is since 1940. You can see a significant drop in the availability of these nutrients in the food we eat. What we noticed in the first 20 months after restoring, or as we were restoring beam, was an increase in manganese and iron availability in the soils by 1,100%. We saw magnesium, calcium, zinc, and copper as well come up in availability, and also nitrogen, phosphorus, and potassium. What we are seeing that in our soils, we're about 8.2 on pH. According to this, on physical chemistry, iron and manganese should have been decreasing. But what we saw was an over 1,100% increase. And this is due to the microbes being able to free that up and make it available for the plant. We're seeing improved crop yields as well. Conventional in our area on cotton production, we get about 1,170 grams of pounds of lint per acre. We're seeing with bean, we were doing 2,472, so over doubling the potential crop. We also saw the same in chili, moving from 10.5 tons to 20.8 in the trials that we had. Now, organic, conventionally, they take a hit on productivity. They are basically conventional agriculture using natural amendments. They have the potential, if they bring the biology back, to be up here and to be even really more organic than what they are right now, using the biology for those nutrients instead of trying to haul them in. This was our first cotton crop in 2010, our conventional with about 168 kilograms of nitrogen per hectare. And this is beam after 1.5 years. About double the productivity, but again, not that great. This is in 2017 when we tried cotton again. This is July 17th. This is August 2nd. And this is August 29th. We had about 6 foot tall cotton. Now, that doesn't mean you have cotton unless you do. In this case, we were growing about 5 bales per acre of cotton, about 2,282 pounds of lint per acre, and about 3,300 pounds of seed per acre. So this is about a $2,000 gross profit, a gross yield on this crop. Our chili as well. And the guys at the experimental farm were waiting for our chili to be done because they know they had never been sprayed. So we didn't use any fertilizers on this or any herbicides or pesticides. We got a corn production project this last summer, and we were moving towards more inoculation or injection of an extract as we planted the corn into the furrow, see just what the potential was. We were injecting at about 1.52 pounds, or roughly almost 2 pounds of compost per acre in 20 gallons at a 20 gallon per acre injection rate. You can see here what the compost extract looked like. In the compost extract that we applied, we were putting about 83.4 million bacteria per square foot and about 10.4 million fungal spores per square foot into this system. And this is kind of what we saw. You can see we had a conventional approach over here on the right, and it repeats across the field, but we were using 256 pounds of nitrogen per acre on this system. This is our other treatment where we reduced that nitrogen by 85%, and that was about 38 pounds of nitrogen. And we were adding that 2 pounds of compost per acre rate as an extract injected into the furrow. And this third treatment was just bean, just the biology, no fertilizers at all. We also had a sub-treatment on this of multi-species cover crops on the left-hand side at each of these fields, and a single species, a triticale cover crop. The multi-species was 7 species. You can see in each treatment, you have a greener corn, even in the one where you've put down 256 pounds of nitrogen. So the multi-species cover crops are much more helpful in making that nitrogen available in the system. So we matched the productivity of the conventional with the beam plus 15% of that nitrogen. And with the beam alone, we only suffered a 6.6% yield drop. Now, this was our first year on this land, and the land was very compacted. Water infiltration rates were over 10 to 11 minutes. The cover crops that we grew didn't grow into the ground. They made a hump on top of the ground. So when you drove over it, it was like driving on a corrugated road. So we did not start with the best of conditions. We saw there was about 40 pounds of nitrogen per acre, and we hauled out in the corn crop 261 pounds. So there was from 180 to 200 pounds of nitrogen that the biology was able to fix and make available to the corn plants. And we saw where in the bean plus 15%, the farmer was able to make $88 more an acre, and the beam alone, about $60 per acre more. Now, I know the cover crop species, the mixed species covers are more expensive, but when you look at it from a different perspective, look, you actually get more productivity, more biomass grown. And if you calculate it on the cost per gram of biomass, it's cheaper to do a 7-way species cover crop mix. So highly recommend, if you do cover crops, move into multi-species so you get more benefit as well in the future. And this is what it looks like on that extract. This was just a small test plot looking to see if we had injected the extract in the furrow, or if we just dribbled it on the top of the soil. You can see significant difference in productivity between these two. So I highly recommend that you inject this extract into the furrow to get it in contact with the seed. So when it germinates, everything's ready to go. I can't go without saying Ian and Diane Haggerty are a big part of why we've moved forward so fast in this arena. They are making great strides. They're doing, I think they started another 20,000 hectares this year, changing the system from a conventional to a biological system. They produce about 3 tons of barley per hectare in a region that gets 1.5 to 1.6. And they've done this just with biology. This is looking at, on the right-hand side, this is the barley, the rhizosphere that is developed. And you can see this seed is barely sprouted, and yet it's putting a significant amount of energy to develop that community. Now, the picture on the left here is after they've harvested the barley. And this is probably the most telling picture I've seen as far as what the biology does in this system. But they got a late fall rain, and in nice, neat rows where they had injected the extract. This is a C4 grass they hadn't seen in 4 decades in that area. So it's the dynamics of the biology in the soil that will move this forward and make it more profitable and much more fun to farm. So I'll kind of stop there and entertain questions.
Wow, hey, I'm just amazed at your ability to package all that information into 30 minutes. That's a real.
Well, you said 30 minutes. That was the Cliff's Note version, okay?
I know, and you've got so much more to offer. Which, and this, thankfully for us, there's some other presentations online if people want to dive deeper into some of that. And I mean, I think I wrote down about a dozen questions here, and there's a few that have come through on the Q&A, but just one of the things we haven't covered in terms of like where this you're talking about carbon partitioning and that kind of stuff is, you know, there's sort of, my understanding is, some of our older understanding was soil carbon is built through the process of decomposition, and that kind of stuff. Whereas I think what you're referring to is the role of root exudates in providing that carbon. So I wonder if you could talk to that and how the kind of microbial, the role of microbiology and stuff in that process and that carbon kind of building thing.
Okay. Yeah, it's you know, it's more than just exudates. It's more than just the biodegradation of material. As you saw, I pointed out that one microbe. It was one of the highest population counts in these soils. It was a microbe that actually produces cellulose in the soil, and cellulose is just plant material. If our microbe, I mean, if you have that microbe, you're going to be building up carbon faster, you know, because that's its home, is what it's building. And instead of respiring that carbon, you know, you're devoting that carbon to, well, new microbes that can function to extract more nutrients out of the soil to feed the plant. It's a system where you're starting to stack enterprises. You increase that net primary productivity on the front end, you know, better photosynthetic efficiency or capacity. That feeds the microbes. The microbes increase in population. They're able to extract more nutrients to funnel back into photosynthesis. It's a nice circular system if we allow it. You know, everything in nature is cycles. And building up that reservoir, I'm not saying ever a static carbon. It has to be, and it's not an amorphous carbon, it's a functional carbon is what we're trying to build up in these soils. A carbon that can mine those nutrients, a carbon that can fix nitrogen out of the atmosphere. We're pretty much, what we've done is just hamstring this system with our practices. And we should work in there and emulate nature, observe what goes on in this system and move forward in the most beneficial way to the soils, because we depend on them for everything we do. We depend on them for the energy, because they're the one that fixed all this energy that we're using right now for our lighting, our refrigeration, our transportation, everything on this planet originates from the microbes. And the quicker we realize this in agriculture and start to promote that microbial community renaissance, so to speak, the better off we're going to be in agriculture. But they make all the decisions for you if you allow them. You don't have to worry about, well, do I need to apply nitrogen? Do I need to apply phosphorus? These decisions are made for you.
A little bit of a get out of the way. And I think Terry McCosker it is that refers to the upward spiral in relation to this, which I really like as a analogy.
Yeah, it's just keep stacking the capabilities of the system on itself, and it just gets better and better and better.
On a related question we had from Peter, one of the criticisms of the potential to build topsoil is the kitchen sink model. More carbon in leads to more carbon out. Your comments about microbial efficiency to debunk this?
Yes, and this is kind of different from most perspectives, because most of the researchers, they see this linear increase, but they're at the very beginning of this curve. They're still working in a destroyed soil. But as you get up to the top, when it starts to plateau, as you get a healthy soil, they'll see that that respiration starts to go down. Now, I've seen 2 research papers that have observed the same thing. They didn't have an explanation for it, but they were in natural environments. So if they're continually working in poor soils in that bacterial dominant low carbon system, yeah, it looks very linear. And if you extrapolate in that way, then you're going to have an error. You have to have the data to support what you see as it leveling out. I've seen it in 4 different experiments I've run. I've seen it in Will Harris's as well. We have a project going in Georgia or in the Southeast US on looking at comparison of 5 AMP branches of adaptive multi-paddock grazing management to 5 conventional. And we're seeing a very high correlation to that carbon use efficiency in the soil, where the conventional is almost double what the AMP is, and the AMP is almost double what a natural ecosystem is. So on respiration. So if these researchers, go ahead, Gwen.
Yeah, can I ask you a question on that? So you're seeing less carbon being respired from the soil. That's
okay. No, relative. No, relative.
Okay.
Now you're seeing more carbon respired. You can see that curve goes up and flattens out.
Yeah.
But I'm talking relative to the amount of carbon that you have available to respire. And
okay.
That's the key.
Okay. So that could be an increase in efficiency of use, but it could also be an increase in the stabilization of that carbon.
Yeah, I would expect that.
Yeah. So and that would work with having more biofilm organism and more fungi, because you have more mucus and mucilage that can stick to that carbon and make it stable, right?
Yeah, all these components working together.
Yeah.
You know, there's no single anything on this planet that works by itself. Everything is in tandem with something else, and we have to figure out how to complement all the processes as farmers.
Yeah. Did you ever looked at the distribution of your carbon in the soil profile?
No.
Okay.
No. This is beginning. Now we have to look and see how that carbon built up. And if you look at a carbon dating on that soil, the older carbon is at the bottom and the newer carbon's at the top. So it's a system that builds on itself.
Mm-hmm.
As you bring back that ability to capture more carbon in the first place and put more into the soil, functional carbon into the soil, you'll start to see soil profiles build up layer by layer by layer. And that's why we have that chronological order in the soil.
Mm.
Awesome, thanks. We've got a mix of questions. We've got some which are kind of broader, and then some that are getting quite specific on the Johnson-Su thing. So I wonder if we do another 5 minutes of broader questions, and then perhaps we might get you to dive and bring Hui-Chun into the conversation to describe, those key principles for getting that Johnson-Su process right. that sound good?
Sure.
Awesome.
You're the boss. You're running this. So.
I've got the God powers.
Yeah, you do.
From Mark here, going from a cash crop with a high input system, how do you suggest transitioning? Cold turkey on nutrient withdrawal or a staged reduction?
You know, I suggest, we were kind of surprised with that corn production dropping 85%. To still see matching the productivity on corn and making more money. So I think you'd be safe at an 85% reduction if you have a quality compost, a quality microbial community that you can put into the system. That's key. I would, if I were you, I would try different treatments. You know, prove it to yourself. Don't go whole hog into this. Have a section where you're trying this out and proving it to yourself and seeing how it works in your system. You know, don't dedicate everything right away. You know, that's this. You didn't get here in one season, and you're not going to get out of here in one season. So it's just, you know, I'd say prove it to yourself. Make sure that the biology that you're putting in is right. Make sure the equipment that you have can get that extract into the furrow and on the seed as well. But I would suggest trying that 85% reduction. You know, we toiled over that. You know, we knew we were moving out of a conventional system on very poor soils, and we were very surprised at matching that conventional rate on this. So that potential exists. You can do that.
Awesome. And a question that I know that lots of people will be thinking, and we hear it a lot in New Zealand, is, you know, New Zealand is very different in terms of soils, in terms of their age, and also their existing organic matter levels, for example, compared to the likes of the States, and particularly the desertified New Mexico area where you live. So
but can you beat 0.5% or less?
Well,
that's what we have.
I don't know anywhere apart from the beach that's that low in New Zealand.
Well, that's where this used to be. It was a beach.
But so considering that context of ours, can we expect to see any different responses to the likes of a high quality beam application compared to, you know, so we've got relatively high organic matter, relatively high biological activity, depending on how the land's been managed. Obviously, soil structure and health kind of varies quite significantly depending on the management and the system that's being applied. But can we expect anything different? Do we need to be aware of? Yeah, I'll leave it at that.
Oh, well, that's the great gray area. We're just getting into this. There's there's more that we don't understand than that we do. So again, try a small test area, prove it to yourself. You know, to see it have that much impact in a very poor soil, I can't help but think that it would improve what you would think would be a good soil. You know, it depends on how that soil has been treated, what legacy chemicals are still existing in that soil. There's a lot of things that we'll have to contend with. You know, the reason I chose that desert soil was no legacy issues. I wanted to see, without any impact of the chemicals that we've applied, the herbicides, the pesticides, what can the system do once you bring the biology back? And it was pretty encouraging to see that quick of an improvement in the soil. So that's the great thing going, that, you know, if you have the right compost, you get it in there, that this system can recover. You know, it's kind of like what we saw on the Crohn's disease. You know, after 24 hours, that person, the change in their life just by restoring that microbiome in their stomach. Kind of think the soils are going to be the same way.
And it's going to be fascinating if we apply the same thinking to the animals' microbiome and stuff that we have in our farms too. There's an untapped opportunity.
Well, yeah, there's incidences of that it helps animals as well. So.
Awesome. Hey, thanks very much for that. I wonder if most of the questions coming in through now, everyone seems to be like they're pretty convinced that your kind of narrative has merit, and they're all asking very practical questions about how to build the reactors and what the best way to apply them and what rates and things like that. So I wonder if you and Hui-Chun can cover off some of your key principles for 5 or 10 minutes there, and then we'll dive into the questions that haven't been answered, if that sounds like a good approach.
Okay. Well, it's a pretty simple system. The materials can pretty much be obtained anywhere. You know, it's just landscape cloth. It's a wire cage made out of 6x6 remesh. There's what, 6 4-inch tubes that are in the center. They're only there for 24 hours though. Just there as a form to build the pallet, and they go through the bottom of the pallet, so you have airflow up through the center. It's key that you stay aerobic. It's key that you do not disturb the material. 70% moisture content is also, that's where fungi do the best. And then worms. Get the worms in. It's usually, the heating phase on this usually only heats for 5-6 days, and then it's done. There's no more heating. Like in windrow composting, every time you turn it, it reheats. So you're disturbing that system. It never really is allowed to progress forward like nature had intended. But having this static system allows those phases, that maturity, to happen in the system. I think that's what's going on. We'll find out. We'll be doing analysis on 10 composts made in 10 different locations across the US here in this coming month. And that will give us an idea how similar or dissimilar these communities are in the compost. So stay tuned. and yeah, we hope to figure this out. You know, it's very complex. Nature doesn't always choose the same microbes to do the same thing. You know, there's many microbes have multi-talents, but nature has her ways of diversity that get it through the process. And you know, we're hoping to learn a little bit more and learn more about, will we be able to tell what's going on in the field as well? You know, can we develop tests that could be field scalable that would tell you, well, you're about here, and this is what the direction you need to go. There's still many, many questions. But the compost, again, seems to be a very good, you know, it's almost got me believing it. But as a scientist, you know, I have to stay 50% skeptic and 50% curious.
Yeah, and anybody who is going to build a new bioreactor right now, I would strongly suggest and recommend that you stick to the instruction. Don't shortcut any steps, because every step is important for a reason. And we made our mistakes, so you don't have to make that mistake. And so, you know, stick to the suggested material, or if you are going to do substitution, don't try something too wild, because again, those materials are picked for a reason, for structure integrity, for air permeability, for able to contain the moisture content. Because a lot of time people say, well, I live in a tropical area, and we have high humidity, we have rain and things like that. So they decided they're not going to cover the top. Well, then they ended up with a few days of dry weather, well, not so much dry weather, but no rain. So they have a top part of the pile dried up. So you have this uneven maturity happen, or the birds come in and start eating the worms, or the seeds get blown in and start germinating. So this is what I always tell people, that throughout the process that you cover the top, and then you keep a 70% moisture content. Whatever you do, don't let it dry out. Whatever you do, don't let it freeze, because this is a biological inoculant incubator. These are living organisms that you are dealing with. They're not going to fare very well in a freezing condition. A majority of them won't. And you are going to bias the species or the entire maturity process. And also that if you let it dry out, you and I don't do well drying out. desiccation does not do a good thing for living organism. So air, don't let it suffocate, because if it gets suffocated at 100% moisture content, that will consider a suffocation. Or that if you substitute certain type of material that would not allow the air infiltration, then it will go anaerobic.
Yeah,
so those are very important.
Yeah, and so there's been you would have seen that some people, there's been, depending on the resources available, people are making small modifications to your original design, and often, those square totes, as we call them, that kind of stuff where they're adapting that. So what are the, and I know Simon and I, when we were doing it, we used a range of different mixes of materials, some chopped, some unchopped, as a bit of an experiment to see which of them would maintain that airflow, as they settled, and what perhaps wouldn't. And so I suppose when we go and actually start looking at them under the microscope, or when Gwen gets her eDNA analysis onto them, we'll hopefully we'll get to see the different impacts of those things. I mean, you've probably already done a lot of that work. But
go ahead.
What's the rules of thumb in terms of how far apart those pipes, the distance to air, I suppose, within it? If people are modifying it, what's the maximum kind of distance to air for any part of the compost?
Okay, what I observed in the research was up to 1 foot is the extent with which oxygen will go into a compost pile. After that, it goes anaerobic. Yep. And that's why the windrow composts have such a problem. And they're turned to re-aerate them, but they go anaerobic within a couple of hours after that. So maximum of 1 foot. We try to keep it about 6 inches for a safety factor.
Yeah.
But
so what you have will be like, if you have a 1 foot bulk, and then from either side you will need to have some kind of a source the air can infiltrate. That's why we have designed the way how the location of those pipes are in the bioreactor. So from all different angles, that there will be air infiltration into the bulk area. And after 24 hours, that you pull those pipes out. And because after 24 hours, that the fungal hyphae has grown through the pile and stabilized the pile.
You can actually see the threads of the hyphae and the actinomycetes. They completely stabilize the pile.
Yeah.
So when you pull the pipe out, you have a nice column.
And so the entire surface area of those columns becomes the wonderful air shaft to aerate without any kind of movement. It just just the air itself permeates through those surface areas.
I think what you said there, the importance of actually soaking that material properly at the start to allow, so there's enough moisture for that really early development and stabilization. Because I think we were a bit nervous. We left our pipes in, I think, for 3 days or something, because we didn't want it. I mean, to be fair, we built excessively tall ones as well because we were a bit greedy. But we had big rebar mesh. I couldn't be bothered cutting it in half. But so how do you tell, I'll reinforce to other people the importance of soaking the material properly beforehand. But what does 70% moisture look like? How do you tell?
Well, in the material that you start with, what we're trying to do is 2 things. Trying to remove all the trash and the soil and the rocks out of the system, so you have a pretty clean product when you're done.
And also, it will be very good to invest a good chipper shredder. So that way that your input material will be shredded into more uniform size. And so when you put things in, that's another thing about it, is that if you just, we've seen people just dump anything into the bioreactor.
You wouldn't believe what we've seen.
And they're chunks, and there are like intact, big straws and things like that. They're going to compost at different rate. And also another part is that when you have all the different bulk density, and when you pull out your pipes, the possibility of premature collapse is there as well, because you have different weight, you have a different density, different shapes, big clumps. smaller packed area and a more air gap area. So, you know, those fungal hyphae are great to hold the smaller particles, but they are still pretty fragile. So if you have those uneven sizes, and on top of that, those larger material, they will transit through the compost 1 year period as well. So, so yeah, invest in a good chipper shredder would be good.
We've got a few questions.
Oh, go ahead.
Oh, sorry, just around any materials that people should be wary of not putting in there. So we have one, like a couple that have come up as like walnut leaves or poultry litter. Those are the two that I just recall seeing on there. So is there anything that we should be wary of not putting in there for any reason? And in terms of that evenness of material, I assume it's still fine to have multiple different materials in there, but you want to try and even out the size of them. Is that the point you're making there?
Well, yeah, when I started with dairy manure, and it was very dense. And so that's why I used wood chips in there, 3/8 inch minus, to change the density of the pile so I could allow that airflow.
Yeah.
Now, I've kind of progressed, and as you saw, moving that decade, going just to leaves. I do just straight alfalfa now because I can get alfalfa that the university is throwing away. And that's the compost I used on that project in Arizona was an alfalfa-based compost. You know, just some bad hay is all right to use as well. I don't suggest the wood chips unless you are trying to have something really dense. Like I think we had some people try chicken offal. The first couple of days was a little ripe, but the pile straightened out. This is a forgiving process. And, you know, as far as the water content, it's pretty forgiving as well. When you have just organic matter in there, the water moves through that pile it doesn't build up like if you had soil in there or clays. You get to pockets where it gets very wet. If it's all organic matter, there's a natural flow of that water through the system, so it never really gets above 70%. So that you keep in mind. Material-wise, the chicken litter is fine. Don't do all chicken litter. Mix in some roughage because it'll be a hot compost. As I say, the heating process is normally about 5-6 days, and then the temperature will go down. It will be through with that heating process, and then you just allow it to go down from there. Keep it all the conditions right, and it, as I say, it's a very forgiving. Watering, definitely put it on a timer.
Yep.
Ask me how I know. Because I can't remember to go and water it every day, and it's about 1.5 gallons. So that'd be what, 6 liters roughly a day is all you need. And I spray it. Right now I've gone to 6 of those what we call a spit spot sprayer that puts it puts a nice mist into the system, and I use 6 of those because each one of those will do about 1/4 gallon a minute, and I just do it a minute a day. And then as long as you keep that, it's pretty much trouble-free. It will progress on its own merit there. Now it will eventually fold in on itself.
Yeah.
You start with about 2,000 pounds of material, of wet material in the bioreactor. You end up with 700 pounds. And so that gives you 350 acres. And let's see, how many hectares would that be? Divide that by 2.47. Do that in your head quickly. But, focus on quality, not quantity, because it takes so little of this to actually get an impact. The cost is only from 25 to 50 cents an acre. And that's a good investment. If nothing else, just the insurance of having your crop do better.
And something not to put in is be like fresh grass clipping. Yeah. They will pack and it will create anaerobic condition.
Best to dry it out first.
Yeah.
I don't see anything wrong with walnut leaves. I could be wrong. I've never done them. I've done many other types of leaves, but you don't see walnut leaves building up to the excess in the environment. So there's something biodegrading somewhere. You might go to an area by walnut trees where it is degrading and get some of that duff, that organic matter that's underneath it, and put that in your bath water as you run the material through. So you're inoculating those with the right microbes to break them down. But tropical leaves, they have waxes that make them pretty impermeable to break down. And if the leaf is broken open, that's why she was saying, invest in a chipper shredder to break those open, and the microbes can degrade it from the inside out. Pine needles as well. As she is saying on grasses are fine, but dry them first and then re-wet them. And they go in the pile, they won't compact down and make a slimy film that pretty much smells.
Anaerobic condition.
Yeah.
Or sawdust. I know a lot of people had asked about sawdust as well. Wood shavings
are great.
From woodworking, that's great. But sawdust, they will pack.
In moderation.
Yeah. Anytime when you're starting to alter any material, it's just like cooking or baking. If you substitute something, you need to be mindful about how would this change that density, how would this change the texture, those things that you put, how would this change the air permeability. Yeah. And that part is very important because it is 100% aerobic composting system.
Cool.
And also,
sorry.
In those instructions, I just want, because other people had asked the questions before and made mistakes before with the assumption, is that the water bath part is very, very important to get the material at the right moisture state. And also pitchfork out is very important because as you're doing the water bath, it's settling out all the sand and rock and dirt out of those organic matter. And when you pitchfork out, it's leaving all that stuff in the water instead of putting into the bioreactor. But as you pitchfork out into a wheelbarrow or whatever container that you have, that you continuously to drain the excess water before you will put that into the bioreactor, because there were people skipping steps and they thought they can use a front-end loader, use the big bucket to pick up all the raw material and then wet it really well, tip the bucket and drain the water good enough, dump it into the bioreactor. You ended up with this really high moisture content. You actually have water coming out of the bottom over the place, and then you ended up with a lot of rock and dirt and sand.
Yeah.
And then at the end, they have this end product look great, but when you look under the microscope, then you will see a lot of mineral in there, and that's indicative of that they did not do the water bath properly, and they do not have the efficacy. When you were talking about putting even at 400 pounds per acre, it's a dusting. They're essentially putting a lot of sand and dust on the land when they're doing 400 pounds per acre. And if you're looking at doing 2 pounds per acre, making extract, then there hardly have any microbial community in the extract, because a lot of that 2 pounds material will be just sand and dirt.
Try it one time this way, and after that, innovate.
Yeah.
You may figure out a better way.
Yeah, and share with everybody.
In fact, there's a high probability you will.
Yeah.
But just to kind of see how this progresses forward, so the observation skills you develop, that if you do try it a different way, then you'll see that you're still going to end up with the same product. We had some people make a 40 by 30 foot bioreactor in central Colorado. They had to put it in a greenhouse because they get so cold there. The compost actually got down to 32 degrees, and they checked, and the worms were still moving around and operating. So,
and they had to build a subfloor, so allow the aeration.
They had a tough time building it.
But it was heck of a time for them to fill it and try to figure it out. But fortunately, they're not discouraged, because the end product that they have is really quite good. And so they're trying to figure out different ways of manage it. to maybe manufacture certain apparatus or structure to make filling the large pile easier. So it's still progressing as far as how to fine tune to make it work that's efficient enough for commercial production.
Yeah, they've already designed and built a trolley that walks along the structure that they're putting the compost in. So the composter, he is sold on this.
Yeah.
You know, he'll be able to make more money with less work than his regular windrow composting operation.
Sounds like a good outcome for anyone there.
He was happy.
I'm just conscious we've got about 10 minutes, and we might sneak a little bit over time, because there's two things that I think a lot of the questions around application. So and I know there's a few key ways. There's early on you were talking about just dusting it across at 400 pounds an acre, but I think you've moved to seed coating and liquid extracts as your primary kind of methods of application. So I wonder if you could just talk us through the pros and cons as you see it of a seed coating, slurry seed coating versus a liquid injection in a furrow, or even a foliar application onto the soil and plant surface, and what the pros and cons of those are, and also what that extraction process needs to be, what kind of pressure you can run through your systems to not harm the biology, and perhaps what kind of rates that we should be thinking of applying this at. And we can convert pounds per acre into kilos per hectare. It's kind of a 1-to-1, so that's all good.
Okay, good. I'm sorry, the US is a little slow. Okay.
Well, it's lucky it's pretty much a 1-to-1, so the numbers still stack up.
The Haggertys are doing all three. They're making an extract and they're spraying it on the seed as they auger it into their seed boxes. They're putting an extract into the furrow as they plant, and they come back, I think, about 2 or 3 weeks after you get germination, and they do a foliar. And they're seeing benefits. And talked to Terry yesterday, and he's seen benefits as well to do this. So, all we did on that one in Arizona was an extract into the furrow. We didn't coat the seed. The seed we actually used on that was stacked. It had antifungal properties on it, and we were able still, even with that, to overwhelm that and be functional. So I would say try it at the very least, injection into that furrow. That's why I showed that picture of, if it was applied topically, it didn't take. And yet in the furrow, we saw over 2.5 times more biomass production on that, having it injected in compared to putting the biology on the top.
And if you are spreading the compost on your land, make sure you have water available right away to water into the soil. Instead, I know this question has been asked many times, and can I just go out there and then just scatter the compost and then wait for the rain? No, again, this is living organism that we're talking about, and you scatter them out there to desiccate and exposed to sunlight, without any water, and then without not allowing them have the capability to go into the soil and start working with the vegetation in the root zone. And so that this is not some kind of a vitamin that you just toss out there. So
still recommend getting it into the soil with the seed.
Yeah.
If any way you can do that is get it into the soil.
Yeah. And so failing that, if you're we've got a huge amount of just permanent pasture, sheep and beef country in New Zealand. If there's in theory, if you can get over and apply it foliar, a few hours before the rain on an overcast day, and that kind of thing, that's probably one of the most cost-effective ways of getting that into the system without having to spend a huge amount of money.
We still need to do the research on that. I know Nicole has seen some benefits, I believe, in Montana. So, again, there's a lot we don't know. I've been kind of focusing in the row crop because I see that area as the most controllable as far as the conditions. And you have the equipment, you know, it's something you do regularly. So we've got a lot to learn. If you graze properly, you can beat what I'm doing, period. You know, the animals are what I'm trying to emulate. So definitely, if you have animals, you can beat what I'm doing.
But if you're growing the pasture and then your land is very poor right now, this will provide the kickstart.
Yeah.
That sounds like a challenge to a few farmers out there.
Well, we just lost you.
Yeah, I'm still here. I've just got issues with my camera, which it does that occasionally.
Okay.
And are we talking, you know, you're saying 2 pounds per acre, 2 kilos per hectare? Is that the kind of rates that you're talking?
Well, you need to take a look at your compost and make an assessment of you know, how many microbes you have. We shot for, remember I told you it was about 83 million bacteria per square foot and about 10 million fungal spores per square foot. So right now, that's all we know. You know, we don't, I did do an experiment looking at using my compost at 1 pound, 2 pound, 5 pounds, and 10 pounds per acre rate. I did not see any difference in productivity.
Well, at least not statistically significant difference. I mean, there is a little bit more for the 2 pounds and 5 pounds, but not substantially more. However, the 10 pounds per acre rate did not
it actually went down.
Do better
in productivity. And the Haggertys have observed this as well as Terry. So you have to be able to assess your compost and do those counts. As far as damage to any of these through the spraying equipment, don't worry about it. The spores, for what they can survive through, it's not an issue. The bacteria as well. These are very resilient organisms, and all these injection systems are pretty much low pressure from what I've seen. Most of them are just fertilizer injection systems, and you're just retrofitting that and putting biology through it.
Just make sure clean it really well.
Yeah.
Yeah. And actually, there's a lot of farmers in Quorum Sense that have are quite experienced with applying biologicals of various sorts through low pressure foliar systems as well. So if people have got follow up questions on that, you know, that Facebook group of Quorum Senses.
Well, they got a choice. They can do it at 25 to 50 cents an acre or whatever the company's charging. And I didn't say that, okay?
I know we'll cut that bit out just to protect your.
I think I'm pretty safe over here in the U.S. for most of them.
Oh, Kiwis travel, David. Look out. We'll get
okay.
Hopefully we'll be getting you both over here sometime soon if you're interested and we get the chance. We'd be
lovely.
Yeah, that would be awesome. I wonder, just you've kind of gone there, but to how do we understand the quality of the end product? And there is quite a few people in New Zealand that is doing these Microscope courses, you know, kind of based on the soil food web stuff, either taught locally here, or there's some people who've actually trained themselves online through Elaine Ingham's courses and that. So, and we have that. You've got the video of the vermicompost versus the Johnson-Su compost that you showed Gwen and I. And I wonder if we could use that as an illustration of, you know, what we're kind of looking for in an end product under a microscope.
Okay.
Is that,
you mean to go to that now?
Yeah, that'd be awesome. Just to wrap it up, just so that, you know, that's probably the piece of the puzzle we haven't quite got to so far. And while you're doing that, we will provide. I'll provide links to everyone to the instructions for building the Johnson-Su. Afterwards, I'll email it out and post it. Maybe post the video. And if anyone's really keen to dive into a bit more detail, there's a Johnson-Su composting Facebook group which is really supportive in that, so you can dive into that too.
Now, this is what my compost looks like when you dilute it 2 grams to 50 milliliters of water. This is wet grams. And what I'm doing here is I'm focusing in and out to see how many microbes are out of my field of view. They're not in focus. I'm focusing back in right now. You can see all these little organisms moving around are bacteria. This is a fungal spore right here, and this is a fungal spore. This is about 50 micrometers across, about 2.5 nanoliters of volume on this slide. This is a hemocytometer that I use to do the analysis. It's relatively simple. Just looking at the biology, there's a spore, there's a spore. I do just the spore counts, then I do the number of microbes. And I shoot for about that 83 million microbes per square foot and the 10 million fungal spores per square foot right now until we know better, because we saw such great response to this system. But it's relatively simple. Here's a spore. Let me stop this here. See, I told you that the spores range from about 2 micrometers, which is said this is 50, up to about 20 to 25 micrometers. And you see this one is one of the larger ones as well. Here's a spore as well. And then over here. So.
And what magnification is that at, Dave?
I think this is about either 200 or 400x. Mine's kind of strange because I have another multiplier because I have a camera mounted so I can take these videos. And I usually do this on every one, so I keep a record of what I'm viewing. But there's about 153 bacteria per grid on this. So, if you're doing a count, you got a very good compost if you can do it that high of count plus the fungal spores as well. And you wanted to see a vermicast that somebody sent me as well.
Yeah, if you it's just a great comparison to show the difference in what a good quality vermicompost can do looks like in terms of diversity. I had one person send me vermicast and they said that it was a great product. And then once I pulled it up, let me see here. You can see there's quite a difference. It'll zoom in here in just a minute, but there's not a lot of biological activity here, and there's very few spores. Here we're really going into that same scale that we're viewing, but you can see very few bacteria. There's only 8 to 10 bacteria per grid on this, compared to 153 to about 200 on mine. And if you do these same dilutions, 2 grams of wet compost to 50 milliliters of water, and then you can start comparing these composts together. It's striking to see, you can see differences. Whereas when you're looking for fungal hyphae, you're not going to see it in this. It's more at this point, the microbes have completely degraded the material and they don't have no more food resources. So what do fungi do? They sporulate. And the bacteria as well, a lot of them will turn to cysts. But it seems like it's that about 83 million bacteria per square foot and 10 million fungal spores. And you look at the diversity on the spores as well, all the different phenotypes. Awesome. The point's not to say that vermicompost isn't useful, because I know there's lots of people getting really great responses to using vermicast and vermiliquid and that kind of stuff. But it was just I think it was just an awesome illustration of in terms of the comparison. So thanks very much for that.
Here's some of the equipment. I'll just go over that real quick. Like a 50 milliliter centrifuge tube, and I use cover slips. Here's the hemocytometer. I use a micropipette, but you can just use a simple pipette. Again, weigh 2 grams into there. Add water to 50 milliliters. Shake it vigorously for 1 minute. This is what the grid on the hemocytometer looks like. So this small blue grid is what you were looking at. That's about 0.25 nanoliters. I'll put about 12 microliters of solution in under, so it will be sucked in to this area where the grid is, and it makes a nice homogeneous film. I use this microscope. I have a camera mounted on it to take the videos. So, and this is some of the spore morphology, what some of them look like, just to kind of give you an idea of what you're looking for as far as spores. So, and you'll see all different kinds. It's a pretty neat system. It's fascinating to go in there and look and see all the organisms. It gives you a good appreciation for the life that's going on in that soil to support you.
Yeah, that's amazing. Because of the connection, I think some of the images are a bit blurry. So we haven't had small tech. Is there a chance that we could have a copy of this presentation?
Yeah.
I'll share that people can. Is that okay?
Yeah, I'll have to find a faster connection. I'll go to university and do it, but I'll try to get you a copy of all this so you can make it available.
Even if it was a PDF without the videos or something.
Oh, it's over 1.5 gig as a PDF.
Oh wow.
I have lots of pictures. I need all the pictures that I can get, okay? Otherwise, people think I'm crazy.
No, it's just there's lots of it's just awesome that you've got the practical on farmer tailored kind of process that you've developed with all the science and data that sits that's sitting alongside it, and we've kind of, I feel like we're really missing that part of the puzzle from an agricultural science perspective. This kind of, how do we apply this kind of microbiological understanding and ecological understandings to an agricultural system? And there's not many people that I've come across in the world that can do it, and you're just doing a fantastic job. It's really exciting.
At our university, I just found out yesterday that some of the soil scientists there figured that cover crops are good for soil. Okay.
Yeah.
Now they're about 2 decades behind farmers.
Yeah.
And every farmer will tell you that.
Yeah, but I mean the opportunities to for farmers to partner with scientists like yourself to do this kind of work and really drive it forward, that's the really exciting opportunity, and that's where
it is.
Queen's just being amazing.
It is so good to see that we can finally work together, and we can start to reinforce what the farmers are seeing. They're already seeing this. Most of them that we talk to, they just don't really have the explanation of why it works, and we're trying to bring that in as best we can.
And I think a lot of the progressive farmers, they have done this successfully in the last 2 or 3 decades. They felt vindicated.
Mm-hmm. We're just trying to catch up to them. They are, the Haggertys are ahead of everybody. And we have others here that are venturing out, experimenting, observing, which is key to this process. You have to be out there and observing. And most academics, they're not out in the field very much. They don't have that opportunity, or they don't take that opportunity to observe and see all that's going on in between. So
it's a very exciting time. Hey, thank you so much for
thank you all
for putting this time. Really appreciate it.
Thank you all for what you guys do as well.
Yeah, cool.
So we all can learn together.
I'm trying to give new depth to the meaning of your organization.
Well, you've got to vindicate the name of the group too, don't you?
Yeah.
But,
might as well check it off, right? While we're there.
Hopefully in 12-18 months' time, we're gonna, Gwen's gonna be able to send you a whole lot of her own data with similar kind of trials, and we can start to triangulate different experiences around the world. It's going to be really exciting.
I love to share what I'm seeing as well, and maybe I'll give you a few ideas of what to look for, so we could kind of recheck and see, well, does it work in New Zealand?
Yeah, that's the question. Any last comment there, Gwen?
I'm just really grateful for you to spend the time with us today, and each time I get to speak to both of you, it's really inspiring, and I just wish we could find a way of working even more together, and just really, really exciting. Thank you.
How about quorum sensing?
Yes.
We're game for that too.
Yeah.
That's how we will move forward. We have to work together because we need all these different eyes on this. That's another thing. Farmers, get into a support group and get with people with like mind and you can, what Gabe Brown does with all his people he associates with, they all try something crazy each year. So you have multiple eyes looking at multiple, at one issue with multiple solutions. And instead of just 45 tries at this, and you're 45 years in this industry, you extrapolate more and more observations that will give us the path forward.
Yeah, you're sharing your observations with other people.
Oh, the speed of learning for someone that's just stepping into this kind of field of thinking, what they can achieve in a year compared to people doing the same thing 5 years ago is just incomparable with the support that are available now. It's just so
Will
exciting.
Harris, Will Harris was 20 years, but he says he can do it in 3 now.
Yeah, 3 to 5 years. That's what most of them say now.
So that experience is critical.
Yeah.
Thanks to the legends like Simon Osborne here on my left, who have been putting in that 20 to 30 years of hard fought knowledge and understanding, so that the rest of us can benefit from it.
What's interesting is we had one person talking to a farmer, and the farmer said he'd been farming for 45 years. And he says, "Oh, wow, you got 45 years of experience." He says, "No, I've got 45 experiences." So that's the difference.
Yeah. Awesome. Hey, thank you again so much, and thanks to everyone that's been on and participating, and thanks for all those questions that have come in. And I'm sorry we didn't. There's probably still another 15 that we didn't address specifically, although I think we covered most of it off. And there is a whole lot more information online that I'll make sure that people are linked up with, so they can follow that up. So.
Well, thank you, Sam, and thank you, Gwen, for putting this all together. We appreciate it.
Thank you all.
Right.
Keep up the good work.
Have a good rest of the day.
Yeah.
Okay, and I'll work on getting a copy of that to you.
Cool. Really appreciate that. Thanks, David.
Okay.
All right. Take care.
See ya.
Take care.
Bye-bye,
Good night,
everyone.
everybody.